Welcome to ASM Live from New Orleans, Louisiana. This is the 115th General Meeting of the American Society for Microbiology, and we're really pleased that you can join us today. We're going to talk about some important, interesting, and sometimes downright surprising discoveries in microbiology. We invite the audience to join in and ask questions. If you have any questions, please go to the microphone and introduce yourself when you get to the microphone. And if you're listening on your computer, please send in your questions at hashtag ASM Live, or you can also do this through the ASM Facebook page. I'm Stanley Malloy, I'm your host today, and we're here this morning with Sarah Cummins and Dr. Jay Lennon from Indiana University. And you work on the skin microbiome, something we think we all have a good idea about our skin because it's the most outward part of our body. But you have some really surprising and interesting findings about the microbiome. Could you give us a brief summary of what you've learned?
Sure, yeah. So in general, our laboratory is interested in microbial biodiversity, and we approach it from an ecological and evolutionary perspective. So we're interested in the processes that shape the structure of microbial communities. And over the last decade, there's been a lot of research that's gone into characterizing the microbial diversity of different types of ecosystems. And we can go in and sample a gram of soil and recover tens of thousands of species. So from an ecological perspective, we're interested in what maintains the diversity of these microbial communities. How can we pack all those species into that relatively small volume of habitat? And so we focus on some of the different ecological and evolutionary strategies that microorganisms have. Often people in microbial ecology are thinking about processes like competition and predation, which actually sometimes can limit what species live in a habitat. So we've kind of relaxed assumptions about the strength of interactions of species and start to think about the ways in which organisms can downregulate their metabolism through a process called dormancy, which is just a reversible state of reduced metabolic activity. So when we've thought about this concept and looked into microbial communities, we know we can maintain higher levels of diversity in communities because of this life history trait. And so when Sarah joined my lab, we started thinking about extending these concepts into the human microbiome and thinking about skin as a habitat where the diversity of our microbiomes may be maintained by this process of dormancy.
So to ask these questions about metabolic activity of any microbial population is a bit tricky. Could you tell us, how do you do this?
Well, thinking about the skin as an ecosystem itself, it's a complex organ with interpersonal and intrapersonal variation. So we have different areas that have different moisture content, oil content, nutrient availability, UV exposure. The skin itself is really hard for microbes to survive on. And living or trying to survive in these harsh environments, they're going to need to develop different life history strategies such as dormancy. So what we did is we sampled different areas of skin as well as sampling people or individuals aged anywhere from infancy to elderly adulthood. And we measured what proportion of the bacteria and microbes isolated from the skin were actively respiring, how many of them were dead, and then how many of them were simply inactive.
How do you do that right on the skin?
What we did was, it seems pretty simple, but we just used cotton swabs and saline, scrubbed the skin vigorously, and then separated the microbial cells from the skin cells just using a filtering technique that actually took longer to develop than we were anticipating. But then after that, we used different dyes that you can see in a fluorescence microscope. So the cells that were respiring would look one color, while the dead cells would look another color. And then the inactive ones would look a third color. So then we could see individually what cell where this one's respiring and this one's dead. And so then we could count up the total number of cells from each metabolic class and determine differences that way.
So this issue of dormancy is really a very interesting idea. The question is, what percentage of the microbes are dormant, And how do you maintain that spot in this niche if you're not metabolically active?
Yeah, so there's two questions. I mean, one idea is, I think what you're getting at is that evolutionarily, it's not a long-term strategy. In order for your fitness to increase, cells need to be actively growing and reproducing. So one thing we were thinking about is getting some numbers on the estimates of how quickly the skin actually turns over and sloughs off. So in infants, that's about every 2 weeks. The skin, that's the turnover time of an infant's skin. But as you age, that increases. And so adults, it's somewhere on the order of 2 months. There are certain people who have skin disorders or inflammatory, like psoriasis, for example, where your entire skin ecosystem is turning over on the order of 1 week. So if you're an individual bacterium and you decide to sleep on the job, then you could potentially lose your place in your habitat.
Right.
So there are potential constraints in how long you could stay metabolically inactive.
So what we found when we did these studies is that overall, the amount of cells that are metabolically active is surprisingly low, somewhere on the scale of 10%. Only about 10% of them are active in breathing and growing, and then the rest were either dormant or dead. And how so many of the cells can be dormant and stay on a specific niche of the skin, the mechanisms of that really aren't known. There's a lot of hypotheses surrounded by it, but the actual techniques that they use to do that is really undiscovered.
Do you imagine, and understand this is really hard to look at, but do you imagine that any given cell might go through bursts of hibernation and rebirth of metabolism, if you would?
Yeah. Yeah, so in contrast to things like, there are other organisms, the life history trait of dormancy has evolved multiple times independently throughout the tree of life. So there's different ways of doing that. And so dormancy in microbes is actually probably pretty different than, for example, classic cases of plant seeds of annual plants or hibernation in mammals. Individuals can go back and forth in between. So an individual bacterium may, like you just said, go through burst, boom, and bust cycles of metabolic activity. I think what's interesting as well is to think, to follow up on Sarah's major point, that is that our skin microbiome performs a lot of valuable functions to us. It defends us against pathogens, it generates vitamins, et cetera. But by and large, again, 90% of the bacteria that are living on our skin at any given point in time aren't necessarily doing anything. They're metabolically inactive. And so the functions that we do benefit from with our skin microbiome is only being we can only attribute that to a very small fraction of the cells. And a lot of people these days are going out into various different ecosystems and using sequencing technologies to catalog who's present in an ecosystem. And often we're making inferences about the presence or absence or relative abundances of these species and assuming that they're contributing directly to some kind of process or function. And with the work that we've done that's been led by Sarah and in other projects that we've done, we often find that there's only a subset of the microbial community that's actually contributing to certain processes or functions and the stability of a microbial community.
It is really interesting. I remember many, many years ago, Norm Pace, when he was really pushing 16S ribosomal RNA to identify populations, said something to the effect of, if you show me the 16S ribosomal RNA profile of any population, I can tell you the metabolism that's going on there at that time. And in fact, you really can't, because it depends on who's asleep on the job and who's doing the job, right?
Yeah, I wouldn't want to put me in an awkward position of going against Norm Pace. I mean, certainly the methods that he's generated have revolutionized the field and have been the cornerstone of a lot of the work that people in microbial biodiversity, we use those techniques. But I think you have to realize that in some ecosystems, not all, there's a large fraction of metabolically inactive cells. And the other thing is that there may be a lot of dead cells that we're picking up. So the nucleic acids, like DNA, are fairly stable molecules in different environments, including the gut, including soil. And in some cases, we have the ability to pick those up with sequencing technology. And it's giving us an impression of who's there or who was there, but it's not telling us necessarily who's contributing to certain active functions.
That wasn't norm pace bashing.
Okay.
When he said that, it seemed like a very reasonable idea. I
Yes,
think it's
yeah.
the amount that we've learned since then. And in fact, part of that is the expansion from just looking at 16S to looking at whole genomes, and now the ability to begin looking at metabolism.
That's right.
So, which fundamentally is core if you care about ecology of any given system, right?
Yes.
So 90% are metabolically inactive. What's the relative amount of dead cells, dead bacterial cells that we have in our microbiome? Is that a large proportion?
Well, it varies. It depends on the person, the habitat, and really the age of the individual. There are a lot of factors that can contribute to those proportions of dead or dormant or the overall inactive pool. But we found a huge variety. Some people would have hardly any dead and a huge amount of dormant, while others would have majority dead and a very little proportional amount that were dormant. But what we found, I think, was about a quarter to a half were dormant, and then about 1 to 2/3 would be around dead.
And what about different parts of the skin microbiome? Is there a different story in different parts of the body?
Yeah, well, what we did is we sampled 3 different areas that vary in moisture content and oil content, so nutrient availability. We measured a sebaceous skin habitat, which has high oil content, high sweat, high nutrients, and we found that that area tended to have a lot higher amount of activity than the other 2 areas, which were, one was a dry skin area that didn't have a lot of sweat or a lot of oil. We found that the dry skin area had the most dormant cells, which makes sense if you think about activity as a direct result of nutrient availability. So the more you have to eat and drink, the more active you're going to be.
You know, if you allow me a little bit of anthropomorphism, it seems like if you were in a part of the skin where you've done what you can do and you know you're going to flake off and disappear, maybe it makes sense at some point. Why keep investing in all of this metabolism when you're on the road to ruin?
Well, I think that can be related to your earlier question about having bursts of dormancy and bursts of activity. There's a lot of species, every domain of bacteria there are species in every domain that can participate in dormancy. And it's not a permanent shut on or shut off. They can respond to small fluctuations in environmental changes and alter their level of metabolism. So, for example, acne is a common skin problem that a lot of people have. And when you take medication for acne, such as antibiotics, the antibiotics are really only going to target the active cells, not the dormant cells. So an individual active cell has the ability to switch to a dormant state to survive. And then when you stop taking the antibiotics, then the dormant cells can actually reestablish their metabolism. So an individual cell can switch from active to dormant and then back to active based on the trial or the span of your treatment.
Let me interrupt for 1 second. I want to remind the audience, please come and ask questions. If you just step up to the microphone, and if you're listening from your computer, please send any questions in through Twitter at #ASMLive. Join in on the conversation, help share asking questions here. So one of the issues that ties back to this idea of turning metabolism back on, you would expect that you could actually do an experiment. So let's say you take a dry part of the body, low nutrient, and you add some oily secretions. Does that reinvigorate metabolism within that region?
I would say theoretically, yes. That's not something that we've done, but in theory, when you put more nutrients in an environment that was lacking in them earlier, then this individual cells should be able to digest that and use that.
If it's really true that they
Yeah.
can come back
Yeah.
to active metabolism,
Yeah.
for sure.
I mean, again, I think these strategies of microorganisms are pretty common, and so there are some logistical challenge and ethical issues of doing these types of experiments with human subjects, but we've done similar types of experiments in other systems, and microorganisms have, in some cases, very rapid generation times and can respond to changes in their environment. I think what's also changing now is our perspective on the other end of the spectrum of the bacterial life history, and that is that they can also survive for prolonged periods of time. In both experiments in our lab and studies elsewhere, for example, in the deep biosphere, which is an ecosystem below in the subsurface ocean, people have estimated that bacteria are turning over once every 50,000 years. So we can think about certain populations of bacteria that are doubling on the order of 10 minutes to the other end of the spectrum where bacteria are doubling once every 50,000 years. So we have a really unique opportunity as microbiologists to study extreme variation in the longevity of biological life.
So I want to just switch gears for 1 minute because what you're talking about is this really very important and interesting interface between microbial ecology and actually looking in detail at microbial physiology and metabolism at single cell level, right? So that's an enormous gap, speaking of gaps in microbes. So how did you get into this? How did you develop the expertise that led you to develop this broad range of activities? This is for you, Jim.
Do we have expertise?
Oh, come on, yeah.
Oh.
Well, I mean, we were talking.
Still developing all of this.
Before the video started, we were talking a little bit how it's hard to predict where your careers are going to take you in questions, right? So, yeah, I think there are some older studies from the late 1990s where people started using some of the dyes that Sarah used, and they measured estimates of viability, and in some cases, the proportion of inactive cells was very large. Getting back to the techniques that we talked about with community sequencing, looking at DNA, we started using not just looking at the DNA of a microbial community, but also what was being transcribed, or RNA. And we mapped those two things on top of one another. There appeared to be a discrepancy. And so whenever there's a discrepancy like that comes up in science, you need to step back for a second and think about what's actually going on. And so that sort of led to some of these ideas that there could be this seed bank or reservoir of inactive microorganisms that could be important for stabilizing function and leading to the persistence of bacteria in beneficial contexts, but also perhaps the persistence of pathogens in less desirable situations.
One of the key things that both of you have mentioned several times is that in all environments, you have this proportion of dormant bacteria. And so let's just choose another environment. In soil, how common is this?
It's very common in soil. That's probably, of the ecosystems that we've surveyed using these types of techniques, again, about 90% of all the individual microbial cells are metabolically inactive in soil at a given point in time. And this is on par with the data that Sarah generated, high levels of metabolic inactivity. There are other ecosystems, for example, the gut, where only 25% of the bacteria in our guts are metabolically inactive. So again, thinking about the high turnover rates or short residence times of your gut, again, this may not be a habitat that favors or selects for a life history strategy of going to sleep. But in the skin or in the soil, the matrix is maybe more stable.
We have a question from the audience.
Yes, we have two questions from the Twitterverse. First, James Kremer asked, do you think that new antibiotics will be developed to target dormant organisms or wake-up drugs and antibiotics?
I think that is probably the most relevant application to this research specifically with the skin microbiome. So right now, antibiotic treatment, there's a lot of drug resistance developing in diseases and pathogens, and it's interesting to consider that we have these antibiotic treatments that target cell wall synthesis products and also translation products, but in essence, the antibiotic treatments are only targeting the ones that are creating those products. So if we have 90% of the population dormant, then it's only targeting 10% of the active cells. And how efficient is that really? How many of the pathogens are we really killing off? And how many of the good bacteria, how many of the commensal species are we killing off? So, those provide us with protection against pathogens and infections, and so we kind of want to keep them. So what I think a great avenue for research and ideas stemming from this are, can we make or develop some sort of molecule that can stimulate or resuscitate only the pathogens? Can we design something that wakes up the bad ones, but not the good ones? So then we can have these antibiotic treatments that'll kill a higher proportion of the pathogens and be more efficient, so we don't need to be taking these antibiotics as long. And then secondly, could we develop antibiotics or medicines that target specifically just the pathogens? Is there some sort of molecule that we can make these antibiotics target instead of cell wall synthesis or translational products? There's a lot of ideas and a lot of room for improvement, I think, and maybe a solution to the issue of all of this multi-drug resistance and this antibiotic resistance that we see now.
Yeah. The second question is from The Log Show, and it is, what was the most difficult process of these experiments that you did, Sarah?
I think developing the methods took the longest. We first didn't realize how hard it would be to get the bacteria off of the skin, the microbes off of the skin, but then once we figured out how to get a high number of them, it was how to separate these individual cells from all the other stuff that's on our skin. Giant skin cells, hair, dust, debris, particles, things like that. You can't look at the individual skin cells if your microscope is occluded with all of these other giant structures relative to the cells. So we tried a lot of things over and over and talked to a lot of people, emailed a lot of people, asked a lot of questions. Nobody really had any good advice. So they were just like, well, good luck and try to figure that out. And I said, oh, okay, well, back to square one about 100 times. But eventually we figured out a way to filter them and let the small microbes through and collect all of the skin cells in bigger pieces. And then we were able to, the rest of that was really easy once we could figure that out.
Persistence, right?
Yeah.
On Sarah's part.
Hi, I'm a fellow, I guess, a micro-tweep of yours, Jay.
Yeah.
My name is Mark Martin from the University of Puget Sound in Tacoma, Washington, and every microbiology educator has always told students that we've got 3 layers deep on our skin.
Yes.
I had an artist friend of mine actually say, when you're holding hands, so are they, which, it really disturbs students, and it's a good thing to think about. I think of the ecology of it. Do you folks think about the dispersal between individuals and the environment itself? It's almost like a cloud, like, I guess, Pigpen almost from the old, Charles Schultz thing.
Yeah. Well, I mean, I think there's a lot of, over the last decade, there's been a lot of interest in the idea of microbial dispersal. And it's certainly just as important. Dormancy allows organisms to basically disperse in time. And the process that you're referring to, Mark, is the spatial dispersion, how microbes move around. I think there have been a number of studies where people have looked at family structures and the similarity of microbial communities among individuals living under the same roof. And there's a higher degree of similarity among those individuals than there are outside of the house. I'm thinking of another example where people looked at individuals in a roller derby tournament. And so they surveyed the community of the skin prior to the match and after the match. And of course, during roller derby, there's a lot of contact. And so the similarity of the microbial community was greater after the people in the roller derby tournament came in contact with one another than before. So I would imagine that dispersal is very important in terms of looking at the composition and perhaps the activity of the skin microbiome.
We have another question from the audience.
Hi, this is Mark Lyles, Auburn University. I'm curious about your thoughts about introducing specific strains into the skin microbiome, specific strains, for example, that are known to inhibit bacterial fungal pathogens, and specifically spore formers. So could you share your thoughts on how you think strains might be used beneficially in that way and how they might affect the skin microbiome?
I think that these dynamic communities that have been growing on our skin since birth have taken a long time to develop, and they probably experience a lot of fluctuations with species and transient species that we pick up or species that we pick up from each other. And I think when a pathogen gets introduced or a fungal community that's problematic, that our skin probably, majority of the time, responds accordingly. It might upset the balance of commensal species that need to be there. So introducing a new strain would definitely shock the community that's there already. And I think there is potential for it to be beneficial, but we probably have to be careful because I don't know how all of the good bacteria could respond to that too. If we introduce a strain that can grow really easily and regardless of its beneficial functions to get rid of the pathogen, you know, it could, are we going to get rid of the good guys too? That's, there's definitely a lot of questions that come to mind, but it's possible. It's definitely something that could be looked into.
I think there are some products now that are being marketed, certain types of creams that, for example, I'm not sure I understand the metabolism underlying this, but you can buy certain types of cream applications that are enriched with ammonia oxidizing bacteria. And that was, I don't understand how that would benefit your micro. I would need to read more about that, but I'm not sure if there's a lot of ammonia production for them to use that as an electrolyte or not. But apparently this idea is catching on within the free market.
It's hard to know if it's like having DNA in your shampoo or if it really is something that's genuinely beneficial.
Okay.
But I think the other part of your answer is so often when people do these grand ecological experiments, there is some outcome that you could not have predicted from the beginning, introducing rabbits into Australia or
Right.
you name it.
Yeah.
And that is where I think that you have to be very careful to have done the clinical trials really thoroughly for something like this. It's also a place where it gets really tricky when you're talking about over-the-counter types of treatments that don't undergo that type of scrutiny. But anyway, we have another question.
Yes, this question is from Janet Rowe of ASM. She asks about the effects of skin, sunlight on the skin, and do you see different communities based on different parts of the skin that have exposure to sunlight?
Yes, in short, yes. So UV exposure is something that some species of bacteria definitely respond to better than others. So areas like our arms and our legs that generally see more sun than the clothed parts of our body, we sampled the arm, and relative to the back or more occluded areas like the back of the knee, we saw that there was generally more dead cells and more dormant cells. Dormant in particular. UV exposure isn't really something that bacteria and microbes like particularly. So I think that intrapersonally, in the areas that have more UV exposure, you're going to see more dormancy or less activity or some sort of life history strategy to cope with that perturbation in their environment. But then also an interesting idea that comes from that is what about people with different amounts of UV exposure in general? People that live near the equator versus people that live in polar environments that don't get as much sun exposure. There's probably a huge amount of variation between those as well. That would be an interesting avenue to explore.
We have a lot of people who tanned in our survey as well. They went to tanning beds, but statistically that didn't really seem to have an effect on the activity of the skin microbiome.
We have another question from the audience.
Yeah, good morning. Cindy Career from Kimberly-Clark. Do you have any idea what the impact on the dormancy state of skin conditions that cause inflammation have on dormancy?
Well, conditions that affect skin inflammation. So inflammation is an immune response.
Yes.
And there have been studies that demonstrated that the skin can differentiate between pathogenic species and commensal species. So there are some commensal species that have either developed less receptors, surface receptors, so they can evade the immune system so they can stay on our skin without being detected. And they can also modify the T cells to target pathogens instead of them. And so if you have a skin disorder that deals with inflammation, then you're going to have either a heightened immune response that the communities either aren't used to dealing with or have been dealing with the entire time, depending if it's not an autoimmune disorder of some sort or a lifetime thing. So I think that if you have a heightened immune system for some reason, if you develop that, then probably a lot more of the commensal species are going to be targeted, and dormancy might be a strategy that they could use to help them survive that. And not only that, but then the immune system might also at first eliminate a lot of the commensal species or pathogenic species, trains, any species. Might eliminate a lot of the initial community, and there's going to need to be a dynamic that settles out, I think, after that as well.
Okay, thank you.
This has been a great conversation. I think it really shows how basic studies on something like the microbiome and an understanding of ecology and metabolism will come together to have a lot of applications in the future in a variety of different areas. But there's still a lot of science to be done before we're there.
Yeah.
And so we really enjoyed talking with you. So again, this is Jay Lennon and Sarah Cummins from Indiana University. I'm your host, Stanley Malloy. We really appreciate you joining us, those of you in the audience here today, and everyone who's joining in online. Come back again and join us for the next version of ASM Live from the 2015 American Society for Microbiology General Meeting.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shower.
It's an unusual room, never seen anything like this.
Anybody who has access to this facility first has to go through an R scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.